Um, all right, let's start. So, um, perhaps the first thing I would like to do is show you some examples of using PCA, okay, on the brain imaging data. Also, I have sent you the like a link to a drive which has the PPT from all the presentations, and I will upload them. Also, many years ago, I had written a paper um, like like Neuroimage had asked me to write a paper about the historical perspective about like this connectivity. So many of the students came and asked me, how did I come up with the idea? So you can read that paper and think it explains perhaps better than I can mention. Um, Also, I recently had a paper in Nature. Um, Someone asked, so I have put that one also. So you can, no pressure. I can't know whether you looked at it or not, but look through the presentation, right? And especially if you have MATLAB, then you can run the code and you should get the results. And I would also suggest you to change the weightings, right? How I added, you can add more or less and see what happens to, ICA or PCA. Okay. All right. So earlier I had showed you this tumor data set, right? In the tumor data set, right, I could see this activation pattern, and when I correlated this, if I took this as my seed voxel, right, you can see this response, right? So this, of course, this is a little noisy because we haven't done any kind of pre-processing. And again, this is because these are tumor cases, transforming them into a template is not very easy, the MNI template, right? So we have to analyze them as the way they are and process them. So what I want to do is show you what would happen if I did a principal component analysis. Okay. You cannot see that well, but what I'm doing is I'm generating the principal component of the whole data, and I'm saving the first 20 components. So this will generate 20 time series, right? Which represents all the time series for this brain, like tens of thousands of time series of the brain, right? Right, so this is done, okay? And what this shows is that the first component accounts for 13% of the variance, the second 8, so it would account for 21, right? Then 27 and so on. So like this is one way to see what percent of the variance do the number of components account for, right? So if I took the first 10, they account for 50%. If I took the next like 20, they account for 65. So that is what I did, right? I have download or I generated the first 20 components, right? So again, this is all data-driven methods. I have no idea what I will get, okay? So um, so what I'll do now is go here, and then display the first component, right? So this is the first component, right? So yeah, data-driven method has some limitation, but just look at the data, right? So I have no idea what the person had done, but I can find out that this is the first component, how it looks like, and then I can do a correlation, right? Now, in this case, what I see is that this is negatively correlated to the motor. Also, what else do you see? There is a lot of edges, right? So maybe this person, like we should have done motion correction, right? And therefore we have edges on this image, right? Everyone is clear so far? And I can play around with this, but you see what I get, right? There is very good motor activity, but they seem to be out of phase, right? So this is negative. Now I will look at the next component, right? The first one was 0, which is the first one. The next is this one, right? You said some same, right? But there is a maybe there is a difference, right? So this one is positively correlated, right? The other one was negatively correlated. This one is positively correlated. So maybe that is one of the limitations of principal component, that although two signals are very similar, it is classifying them as two different signals, right? Let's look at the third one. Okay. This pattern looks different, so let's see. Okay. Hard to say, mostly on the edges, right? And some random voxels. Mostly around the edges, I would say, or this one, right? Um, right, so this is similar. Let me look at this one because it has more fluctuations, like some fluctuations, whether it has any meaning. Maybe not, right? right? Again, so these are what okay, so in this case, this is what I would call noise components, right? You see something, but because I used every voxel both within the brain as well as outside the brain, some noise component is showing up, right? It's not related to any physiology or anatomy. Um, Lastly, I will try the 20th component, right? Okay, so again, so this would be just noise, right? We should not see any pattern. Okay. So this is a person who had a brain tumor. So in addition to motor activity, a language task was done. Okay. They performed, as I had said, it is similar to finger tapping, but um, they would have to generate words quietly starting with a particular alphabet. Like it could be like 20 seconds of rest, then they would be given like letter S, then they would have to generate as many verbs starting with S for 20 seconds, then they would stop, then maybe another letter like B or like random letters would be given and they would have to generate. Okay. So let's see. What happened? Okay. So we are now computing the principal component, right? So for motor one, the first one was, does anyone remember how much it was? Was about 13%, right? This one is about 9%. And the first 10 had 49. This one is 47. First 20 was 65. This is 64, right? Now let us look at the language. Okay, this is how the first component looks like, right? So maybe that is language. Right? So This is the activity. And again, with language, it is more complex than finger tapping, slightly more complex, because if I asked you, let's say you're in the scanner, and I asked you to generate words or verbs, after I say stop, it may not be as easy to stop, right? So the response is a little more complex. Now let us look at the second component. Not really, right? It's also like that. And again, because I have taken everything, right, outside the brain, inside the brain, there is these voxels, which is on the edge, right? Let's see if there is something we can see which is a little, maybe it has a little more temporal structure. Let's see what we get. Hard to say, right? So it's around the ventricles. So most likely noise, right? Because this matches this pattern, right? So you can see how this method can be very useful, right? You perform this task, you have no idea, right? You told the subject to do some task, but you have no idea whether, well, first of all, whether they were able to do the task and whether you get a response. So yes, you can use and you should use GLM, but you can also use this data-driven methods, to see how the data looks like, right? And the other option would be, now that you know that these are the noise sources, these could become your predictors of no interest if you do GLM. And so you would get much better activation, okay? Okay, so this data also has a resting state data. So let me quickly, yeah. Predictors of GLM are the correlated patches or the so the noise sources would be PONI, predictors of no interest, right? So it would be nuisance variable, right? It would not be predictors of interest. You could use the first or the second as predictors of interest, but not the later ones, right? Okay, so the resting state data was a little longer. Okay, the first one is only 6%, the second 0.5, 0.2, right? So the first 10, oh, sorry, the first one accounts for 67%. That is, so just by looking at that, it would give me a little suspicion that when you have tens of thousands of voxel, what could be a signal which can account for 67%? So let's look at the data. Okay, what does it tell us? Hmm? No, where is most of the activation? Right, so if it's around the edge, what does it mean? Yeah, so this person moved ahead. Mm, no, it's on both sides, right? This side as well as this side, right? So this would mean that this person moved ahead, right? Um, so what to do in this case? You have to correct for head motion, take those 6 parameters and make it signals of no interest, right? And then do like redo the analysis. Okay, so that was the first component. Let's look at the second component. Actually, because this person moved so much, the other ones, I would be surprised if we can find anything, right? Okay, this is also all on the edge. Let's look at the third one. What could that be, right? Let's find out. Can anyone guess something? Make a guess, someone? Surya, what do you think this is? The red, the high frequency. Hmm. Hmm. So it is from this near the ventricles, right? The ventricles has like the CSF pulsation. So that is why you see this. Okay. Any question? Everything is clear? Okay. All right. Um, okay, so this was not a very good resting state subject, but it still teaches you something, like by doing simple commands, like looking at principal components, you can find these trends, right? The different components, and you can find noise sources, right? So if it's motion, you must do motion. Actually, you should do motion before you do any steps. But especially in this case, you have to correct for motion. And when you have fluctuations like this in the ventricles, this could also be used to remove any kind of noise sources from the ventricles. Okay. How does the CSF pulse? Um, so this is where like temporal filtering helps, right? By using a temporal filter, we can get rid of the high frequency one like this one. Yeah. 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 Right. 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 That's a very good question, which I don't know the answer to, because some people say that we should look at signal till 90% of the variance is explained. Some people think, oh, it should be 95. Some think it should be maybe 75 is enough, right? So it depends. Look at the, if you're doing EEG, look at the literature and see what other peoples have used. Similarly for fMRI, for like um, I can tell you what people use for fMRI, and even in fMRI there's a like a large variation in the literature, right? So it depends on what the question is. The other thing I would like to point is the the first component doesn't always mean it is head motion. It depends how much it is, and with principal component, it explain the variance. So if the task has good response, like large response, then the task will be the first one, like as we saw for language and motor, right? But if those subject had moved, then maybe motion would have been the first component. And that is also like a limitation with PCA and also ICA, because you can make a guess as to what the order should be, but that doesn't mean it will be that way, right? In some cases, the stimulus response would be the first one, but in other subject, it could be that the stimulus could be a second or third, right? Okay, I hope you have a slightly better idea. Okay. Again, you know, um, I have like I have worked on this for so long, and so maybe something which is very intuitive to me may not be to you, but if you ask, then I'll try to answer. Um, Okay, I will give. Right. So okay. So what we will do is first, as always, set up and do the data loading, and then mount it on the Google Drive, um, and then we will perform ICA, and then we will look at the time series, also look at the frequency component. Um, because I have said, both for PCA and ICA, in some cases it is used to identify activation, used in seed voxels, but in many cases it is also used to clean the signal, right? And then we will use the UO Atlas. Okay, I will just check whether my network is working. Has anyone been able to go through this? How is the internet network at your dorm? Good? Okay. Yeah, so in this one, we are using a different ICA, CAN ICA from Nylon, and I haven't used from FSL because just this FSL loading up takes up so much time that I thought like we should use this one. would any of you use PCA over ICA for some specific condition? Yeah. Okay. Right. Yeah. So if it's a simple task, or if you know it's like blog design or something, maybe it can be used, right? Or if it is important for you to know how much variance a task is, accounting for, then maybe PCA. Again, you can sort of find those things with ICA, but it takes a little time. Hmm. Right, in this case, we are just using the MSc subject 1. Okay. So this ICA is called canonical ICA, which is um, a good implementation, right? It's like, has any of you heard of canonical correlation analysis? So remember the example I was giving you, like about your studying habits versus the grades that you get. So with canonical correlation, it will it will find a linear combination at both ends, right? And it will find which is the optimal one. Like as I was saying that, if the homework is point like 40% of the grade, midterm is 30%, and final is 30%. So with canonical correlation, these kind of relation can be determined. Okay. Right. So these are the components. This is the number of components you want. This is the random state 42. What that does is, because with ICA, because it's a generative model, each time you run ICA, it sets up new initial condition, right? By doing this random state, you're starting at the same each time, right? It's like in MATLAB, the equivalent is like where you put the random number so that it would start at the same initial condition. Again, it would be good maybe for others to also try, right? Oh, right. So then Once the 20 components are generated, we would like to see them. Right, in this case, there are 20 components. Is it running? Oh, that is why. Okay. Okay. Okay, let me use my cell phone. Oh, although if I use my cell phone, I may have to redo everything. Okay, let's try. It ran through it. Maybe I have to go back to the top. Okay, let me just check. Is anyone else having? Right. Okay. Hmm. Uh, let me bring. Just give me a second. Oh, it worked. I think it worked. Okay. Now load the data. Oh, maybe it didn't work. Okay. 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 So this one we are doing resting state, right? So then again, please go through that because I'm going to ask some of you to come and show me how you did. Okay. Then is to do the, so this is doing the ICA. Here n is the number of components. You can change the number of components and see what happens. Also like this can ICA was like faster rather than installing FSL and running it. Has anyone used ICA? Okay. I think this is the, it takes like maybe like 5 minutes. Hmm? Oh, for you? Okay. Mine is already 1 minute, 20 seconds. Okay. So it did this. And now we display the 20 components. Do you have plans to visit some place over the weekend? Yeah? Okay. What happened? So are you, right? Okay. How how do you compare Prayagraj to Kerala? Excuse me. Someone told me you're from Kerala. Okay. How would you compare? Oh, okay. We're in Africa. Botswana. Okay. Okay. You know who told me you are from Kerala? I'll tell you later. Dr. Kar. Okay, so these are the components, right? This is the first one. near the cerebellum. This is the second one. Again, this is the third one, which is from, right? I will let you guess which one. This is fourth, like maybe medial frontal lobe. This is fifth. This is the default mode network. This is seventh, eight, perhaps head motion, 9 or 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. So my favorite network, I can't seem to find, right? The sensory motor is not so clear. But that is okay. This is one subject. So if there is motion, if there is any other factor, then those variants is going to play a role, and therefore it may be that you won't see specific networks, right? But again, I think this is what I'm trying to say, that this is during rest, right? The fact that the brain is connected in such fashion is quite amazing, right? Because with task activation, right, the assumption has been that during baseline is just noise. There is no information. And again, the main assumption that resting state has like suggested to task activation is that not that task activation is bad or anything. It's just that the baseline, there is information in the baseline condition during rest, and that is not random noise, and it has it is functionally related. Also, it has a temporal structure, right? It is autoregressive kind of noise. So the next thing we would like to try is to compare it, look at each network, extract the time series from this network. Right, so it will find the time series, right? So yeah, yeah, right, because now you are saying you're forcing the ICA to run and find 50 spatial patterns or temporal patterns, right? So if maybe try from 20 to 25, right? But if you have a large number, right? What would be the analogy? So the analogy may be if I took everyone's photo of the face, right? And it was transformed such that all the faces were matched. And then if I have like hundreds of images, and then I did a principal component or independent component, I can find features. I can find eyes, I can find like nose, lips, and so on. But if I put in the number as very large, then I will get left eye separate, right eye separate. And maybe my guess is that is what has happened in your case. that is one suggestion. The other thing I would suggest is that run again and see if there is any variation. Yeah, run again and see if it's identical. So here for the 20 networks, we averaged the time series. So now we have 2,818 time points. Now we are looking at their like temporal components. Uh, that's a good question. What do you think? So he's asking with PCA, right? The PCA time series, they are by definition orthogonal, right? So the correlation between the time series would be 0. In ICA, they're independent, right? So you should check what the value is, right? Maybe use it as for your homework and see what do you get, right? So in this case, it's saying that the number of component likely noise based on is 4. And here, what we are looking is for each component, we are also looking at the time and the frequency domain analysis, right? So this one is component 1, maybe noise from power spectrum analysis, because this is how the time points look like, and this is how the frequency component look like, right? And this one, maybe because it has large peak around here, maybe it is due to respiration, but we would have to look more carefully, right? Hmm? Right. Yeah. Yeah. This is the first component. This one is again based on the location. And again, in this case, um, again, we should have done linear trend, like detrended the data before running. But I want to also show you what happens when we don't correct for anything and run through, right? Again, in this case, you have such a large magnitude at the higher frequency. That is why, like, I would be suspicious as to what is happening, right? So this is so high frequency noise here, right? So high frequency, that is why I think this is noise. Again, this is also noise because of the high frequency. Okay. Right, so this is another component. Again, this is, we just have to take a look and then decide whether it is noise or something else, right? Again, because of these parameters, because it's so high, right? The high frequency component. right? So this is, we see correlation along this direction, and we think this is real because there is power in the low frequency range, and in the high frequency range there is not much power, right? So that is why we think it is signal, but this trend makes it a little difficult. This one also, we think this is connected based on the location and the frequency spectrum, also because the data hasn't been, like, maybe this is a little bit of motion, head motion, right? This one perhaps is the default mode network. This, and this two region, and this is how the time series looks like, and most of the power is in the low frequency, right? The same with here, right? So again, these are these fluctuations. The most of the power is in the low frequency, some, right? Very little in the higher frequency. Similarly here, this is the signal, this is the frequency spectrum, right? Again, with resting state, currently most of the signal that we think we get is from the low frequency range, right? But because the time series looks a little funny, like it's initially the noise, like with time the standard deviation seems to go high. That is why we have to be more like careful as to what we are observing, right? This one looks like a good time series, and the frequency spectrum is also clear, right? Um, like this one perhaps is the language region, right? Like, because the time series doesn't look stationary, that's why we have to look through them. And this is, again, the idea is we should be detrending our data and pre-processing our data. Sometime like filtering is not needed, but at the same time detrending and other things should be done. So yesterday I had spoken that Thomas Heo has developed this atlas using clustering method, where they have developed a hierarchical clustering method where they can find like 7 or 13 networks. The UO7 is very widely used, and so here we are trying to look at the 17 UO17 networks. Right, and then we use this data to generate the 17 networks, right? So these are the 17 networks that we obtain. Right, so again, this is a different way to analyze the data, right? So EO has developed this clusters, and each of them belong to different networks, right? And we can take these regions and perform correlation analysis to find the temporal structure that is in the data, right? So that is done here, right? And then we are basically loading the data and looking at the connectivity pattern, right? So again, this is the Geo Atlas, and this is looking at the atlas on the data, right? And so now what we will do is, since we know that these are different regions, we are going to take our data, take those specific regions, and then correlate. Right? So we would correlate and then find this, and this will tell us the pattern, right? So we have 20 components and we have 7 networks. And therefore here we can show how each of these networks are correlated to the 7 networks, right? So for the first Geo network, okay, this has 20 components, this has 7 components, right? So for the first network, component 3 and component 10 have high correlation, right? For the second Geo network, component 7 has positive correlation. For the third one, component 15 has. For fourth one, component 17 has. For fifth one, 18, right? For sixth, 8 and 15, and then for seventh it is 4 and 6. So this is another way to represent the data, right? Right, so this is showing that the third component has the most correlation with Geo's visual network, right? Then the fourth component has maximum correlation with the default mode network. Okay, sixth has maximal correlation also with the default mode network. Actually, the default mode network in this data set seem to have been split. Like generally in the default mode network, this one is also present here, but somehow in this data set, it seems to have been split, right? Then the 8th component seems to have maximum correlation with the frontoparietal network, according to EO, right? And then component 10 has maximum correlation with visual, component 15 with dorsal attention, 17 with motor, okay, 18 with limbic. So this is another way, right, to display and identify networks, right? With ICA, we found, let's say, 20 components, but how can we compare, right? We can't, if we use this data and classify, then it won't be fair, right? Because it would be circular also, right? So what is done is that we used another data set. So your atlas is obtained from 1,000 subjects, right? And so by looking at what we have done and showing the components and comparing with that, it is a different way to like quantify the reliability of these network, right? That is one. Also, this is done on a single subject basis. It's only one subject we can find these network. Also, it tells us how much overlap there is, right? So is every, yeah, yeah,
You already mentioned it's a single subject.
right.
Is it possible if I process the data for n number of subjects
Uh-huh.
and then I see the heat map that is generated for the components 1 to 10.
Right,
If I do it for within components between subjects
right.
for with the heat map, will that be the reliability measure that if I am trying to do a like not a group analysis, but if I have to do a for a particular population?
Mm,
So,
So,
so basically, within the PC one,
right,
first principal component,
yeah,
if I do the heat map for 25 subjects.
okay,
So
right,
basically, I'm trying to see the how how each of the principal components is
right.
correlated with each other for 25 participants. Is that a reliable way to look at the similarity or the differences across subjects?
Right.
Suppose all 25 come from same disease, but obviously they wouldn't have the same symptoms or same
Right.
problems. So is that a reliable?
Um, okay, so um, let's first talk about healthy subjects, and then we can ask about disease subject. So, does anyone would be willing to answer, right? If we use 25 subject, would we get similar pattern, right? Like this, or will we get different pattern? That's the question you're asking. So I would say that with 25 subject, if they are healthy subject, it should slowly look more and more like the UO network, because the UO's network is generated using 1,000 subjects, right? Now, the question is more difficult. Let's say um, they were schizophrenic. Just as an example, let's say you scan 25 schizophrenics. Should they look like this? Maybe, may not be, right? Because maybe in, just as an example, let's say in schizophrenia, the motor and the visual was one network, just as an example, right? If the motor and visual is one network, then technically we won't find a good overlap by comparing with your network. And that is one of the challenges with ICA, right? With ICA, what are the assumptions we have made? Anyone? What assumptions are we making in ICA with this network? Good, right? So the first assumption is that each of these spatial components are independent of each other. Now, if the visual and motor are not, then that violates the assumption, right? So then how to analyze, right? Should we So that is like a challenge in this area, like in connectivity, is to, should we look at resting state for schizophrenia separately and healthy subjects separately, and then compare, or should we combine both resting state healthy and resting state schizophrenia as one large group, find the components, and then go back and see individually, right? Again, I can argue for either way, right? But each one has its own limitation, right? If you combine them, you're getting something in between, right? Which may not be even true, right? Um, so those are some of the things we like, um, some of the limitations. As long as we know, I feel that you are okay, right? That is one. The other thing I would say is that if, like the way we said, um, for going back to schizophrenia, if these networks are connected, what we could do is we can find regions of interest based on each of these blobs, and then do a time series correlation, right? So those, instead of 20 by 7, they would be 400 by 400. So that gives you more uh, specificity, right? Because you are looking at not network. Well, you're looking at network, but you are looking at also sub-regions within the network. So you can find how the networks are affected, but more precisely, you can find how the sub-regions of the network has been affected, right? Any question? Yeah.
Suppose that you have 5 subjects, 10 subjects,
Okay.
5 female, 5 male.
Okay.
So the female
Uh,
is like investing.
right.
Maybe I want to forget agriculture, transfer from 4D
Right.
to 3D.
Right.
Then for the female, in each subject, I'll tell you to stack each other. So instead of having 12 subjects,
Right.
we have only 5 subjects which are stuck together.
Okay.
The same applies for the female as well. So I'm going to like this 5 subjects which has been stuck together for female, 5 subjects for male.
Okay.
Then I want to do this one. I want to do an space of the 7 network.
Mm-hmm.
Is that possible?
Yeah.
Instead of using 4D,
Mm-hmm.
I'm using 3D, which has been stuck together.
So, okay, if I understand you correctly, what you're saying is you would do ICA and basically generate similar networks, five of them for 5 males, and another 5 maps like this for 5 females, and then you want to do a comparison. What is the difference between? Yeah, that is how. So this is actually the way you described is, um, yeah, people are doing that way also. So you are converging different, right? So what you are doing is, yeah, so what you are doing is something like the following, right? You are looking, you're taking each map, color coding it based on the component, and then so component 1 would be 1, component 2 would be 2, 3 all the way till 20. So that would be subject 1. The IC component for the second subject would be the same way, and so on. That is a good idea, and you can certainly do that. The only thing you have to remember is that make sure that they match, that component 6 for all the subject should be this one, right? Or component 5 for all the 10 subject must be looking like this one, right? Only then can you compare. With ICA, because it's based on trying to look at the variance and independence, in one subject this may be component 4, but in other subject it could be component 5, right? So as long as you remember that, there are some simple ways in which you can order them accordingly, and then you can combine and do a test. Any other question? Okay. All right. So let's try. Okay, let's try. We still have half an hour. So all of you were able to regenerate this? All of you? Could you try the second data set? Mm, right. Uh, right, right. Right. So if you have two groups, we would like do all these steps, right? And then basically order them according to like some hierarchy, where we would use, okay, the first one is visual, the second one is motor, the third one is default mode, the fourth one is, and so on. Once you order them accordingly, then you can look at the underlying statistics of these values and then do a z-score and compare them, right? So that is one way. Done? How does it look? If you have, does your computer have power? Your computer has, you can come here and try, sure. Oh, come this side. Huh? Oh, okay, okay. Come to this side.
Okay, so this is the ICA components.
Okay, okay, so now this is the first component. This is the default mode network is that, so a small part is missing. for the default mode. Go down. So this one is, what is component 3? Most likely the sensory motor. Go down. So these are the different networks. This is the second subject.
Yeah.
It also has this trend.
So, yeah. So again, the time series is much more like bigger. This one is the default one.
Right. Okay, that's good. Has everyone been able to get this one?
So, the issue I was facing is, in normal or ruling data, there's a code which says functional find LIN MNI is equals to F data already pre-processed from that folder. But then within the drive, within the data,
Yeah.
yeah,
So
it's already
yeah, you just have to change the path here from already pre-processed to data pre-processed only.
So try that. Also, the other thing you could do, you're saying something? Can you just highlight the line? Right, so he has highlighted what needs to be changed, right? So that is one option. The other option is it would take 30 minutes, but you could run or you could process the second subject data and then copy it in that directory, then you would be able to run also. Okay, so that is one. The other thing is that it would be interesting to try the task data, like the first run, right? The functional task data, and see what do you get. Maybe you can, do you want to try the task data, the first run, the finger tapping, the like again, just do the second subject first, but in the meantime, Shreyans will show you how to do the task data for the first subject.
No, okay, this extension will work. So have you pre-processed the data yesterday for the second subject? So it must be stored in that directory. Still not working. Okay, I'll check after this. It's somewhere around 5 minutes. Maybe 7. Okay, done. The image to display, it will take some. So subject to, yeah, but why not visible? Why it is why coming out as this? Oh, okay. Rest analysis. Okay, yeah. Yeah, not the task one. Okay, then, okay, I guess for the first subject, I do have the task. Okay, the motor one.
I was
No, it will work.
maybe 20. It's too.
Just checking the path once more time. Okay.
So in this case that he is running, the data is not converging, so he has used 20 components, and we had 804 time points. So it could be that the number of time points is less. That is one possibility. The other is that this was a finger tapping or motor paradigm. So like 20 components maybe is too high, and we should reduce it to maybe 10 or so.
10.
So it finished.
Mhm.
Okay. So in this, right, so in this case, the result is quite interesting because like we have to figure out, but in this case, what it looks like is that one component is representing the left motor cortex associated with right hand or maybe right foot, right? This go down. Yeah, so this one is associated with the left hand, right? So it could be that because Shreyans gave 20 components, ICA, although it was a single component, extracted into, because the temporal patterns were different. Remember, like it wasn't both. One was left hand, the other one was right hand. So temporally they were different, right? So maybe that is why it showed it as two different components. So we have to figure that out. Okay. Again, these are cerebellum, and again, can anyone guess what this one is? It could be motor, but it could be maybe with tongue. Like there was a task called tongue wiggle, right? It could be that, right? But we would have to check. So those of you who haven't been able to get the second subject, Shreyans can help you. Thank you. I think I'll switch off. Maybe